Hello everyone. In this video we're going to talk about blood pathology, and blood pathology generally means different kind of disease associated or pathogen that cause diseases that are related to the blood. So let's go into the presentation. So in the introduction, a blood test is a lab analysis performed on a blood sample that is usually extracted from a vein in the arm using a needle or via finger prick. So what we are trying to say here is that a blood test in blood pathology is just like any other pathology test is performed on a sample, a biological sample that is taken from a blood. So different type of blood tests include biochemical analysis, molecular analysis, and cellular analysis. So the objective of a blood test is to analyze blood composition for prevention of blood-related diseases. So the reason we do blood tests, for instance, these days we do blood testing in relation to COVID-19. We do that to analyze the blood and to see if there are any diseases in the patient that can be identified using a sample from the blood. So what are the different processes for blood sample collection? So blood sample collection is performed using injection. So blood is drawn from the vein, usually from the inside of the elbow or from the back side of the hand. So you can do it near the elbow or from the back side of the hand or the palm. So the different tests that are included in a blood test, different kind of blood tests are complete blood count, blood culture, blood film, platelet count, glucose test for diabetes, or Wedel test, ELISA, ammonium ions. So what you can do with in a blood test, you can actually do that for to check the complete blood count, or you can also check your vitamin levels or the presence of any other harmful pathogens. You can count your platelets in a blood test as well. And the blood test results can be different based on the type of the test. For instance, for a blood count test, it can tell you the number of white blood cells, or the red blood cells, hemoglobins, and the platelets. The blood culture test can tell you the presence or absence of microbes, pathogenic microbes. The platelet count can tell you the number of platelets in the blood. The glucose test is usually for to check for diabetes. So the result for normal blood, so if we assume that if we have taken a sample of blood from a patient, and we so what are the different kind of results that would tell us that no harmful pathogens are present in the blood. So for Wedel test, if we get a ratio of immunoglobulin M to 1 to 60, or immunoglobulin G ratio of 1 to 40, then that is normal. For ELISA, if there is no antigen present, and for ammonium ion, if there are no ammonium ion present, or if it's a vitamin test, then the normal test would be normal level of vitamin. For instance, if it's vitamin B, then vitamin B or E. So in conclusion, we can say that any abnormal results means that a patient has a blood disorder, but if the levels are under in the range of what's the expected values, then that means there are no abnormalities. Thank you for watching the video. If you guys have any more questions, please feel free to ask them.